Are you seeing it?
Good afternoon, colleagues. It gives me great pleasure to welcome you to session number 10 of the African Society of Human Genetics meeting. We've had a fabulous few days, and this is the penultimate session. And the plenary today will be given by Professor Solomon Ofori-Akwa. So just to give you a brief introduction, I know he's no stranger to many of you, but it is important that you get a bit more information. So he is the Dean of the School of Biomedical and Allied Health Sciences at the University of Ghana, and also Associate Professor of Medicine and Human Genetics, and the Director of the Center for Translational and International Hematology at the University of Pittsburgh in the US. His research interests are in the molecular pathogenesis, genetics, and therapy of acute complications of sickle cell disease, with a focus on the lungs. He developed the first mouse model for acute chest syndrome, and he has active research programs in the US and in Ghana, focused respectively on preclinical studies using transgenic mouse models and genetic determinants of clinical phenotypes, studying large patient cohorts in Ghana, with collaborative sites in Cameroon, Nigeria, South Africa, Tanzania, and the US. So Solomon is the center leader of the West African Genetic Medicine Center, and as center leader, he is spearheading efforts to train the first cohort of postgraduate genetic counselors and medical genetic scientists in West Africa, reflecting his continued efforts to promote all aspects of biomedical science in the region. He's also director and principal investigator of the Sickle Cell Disease Genomics Network of Africa, called SickleGen Africa. So today he's going to talk to us about the future of genetics of sickle cell disease, but also looking back at the past. So Solomon, thank you very much, and the stage is now yours.
Thank you, Michelle. Thank you very much. I'd like to thank the organizers for inviting me to present. The title is very interesting, The Future of Sickle Cell Disease. But before I get into the talk, I want us to spend a minute to remember a very dear colleague of ours, Paul Frenet, who passed away earlier this year. Paul was a brilliant scientist, a very fair-minded reviewer. I served with Paul on the hematology study section of the NIH. Among the many accomplishments Paul gave to us in the field, is shown on the cartoon to your right, was Paul's work that first demonstrated that neutrophils adhere to the intima of blood vessels, where they then trap erythrocytes, sickle erythrocytes, to form vaso-occlusive plugs that we all read and talk about. I'm sure some of you have used that cartoon to my right without necessarily knowing that it was Paul's work. And so we miss him dearly, and we hope to learn from his genteel approach to peer review. So thank you for allowing me to share with you a few moments remembering Paul Frenet. So now the task I've been given this afternoon here in Accra, is the genetics of sickle cell disease. And I have put on the far top right the Adinkra symbol called Sankofa. Sankofa in the Akan language in Ghana literally means go back and fetch. And you can see it symbolizes a bird reaching back and grabbing an egg. Most people probably don't know that this symbol is actually not about the past, but it's about the future. It's about the bird picking an egg, which is going to hatch yet another bird. And in the Ghanaian traditional military circles, the idea is to learn from the past to prepare the next war moving forward. So it's a very interesting title, of Future of Sickle Cell Disease. And I want us to look back to learn from the past to advance forward. I want to start in 1977 when the sickle cell mutation was actually sequenced. This is it was actually a paper in JBC by Morata et al. This was the same year that Frederick Sanger, Maxim Gilbert basically demonstrate a very simple methodology of sequencing the DNA. The following year, 1978, is when the first globin switching meeting occurred. And throughout this talk, I would highlight how our focus on understanding the molecular mechanisms that control the switch of gamma to beta globin has really been the epicenter of genetics of sickle cell disease. That same year is when W.Y. Kahn and Dozy discovered the very first human RFLP, restriction fragment length polymorphism. And sickle cell played a key role in that because they discovered individuals with sickle cell disease had a different length polymorphism from those who don't have sickle cell disease. The RFLPs that Kahn and Dozy discovered led to extensive use of RFLPs to study the chromosomes of individual sickle cell disease. And in 1984, there were pair publication PNAS that identified 4 different types of haplotypes. And again, this dominated our study of the genetics of sickle cell disease for several years. We're all familiar with the Human Genome Project, which spanned 1990 to 2003. And this cartoon I'm showing you really was inspired by a very brilliant review published by Francis Collins in Nature at the time, stopping at 2003. Now, 2007 is when Swilly Finn, working at King's College London, maybe she was at Oxford at the time actually, discovered the quantitative trait locus BCL11A, and which really was a follow-up of our efforts to understand globin switching. 2012 is when H3 Africa was launched, and H3 Africa has really been a major landmark in the expanding genomic studies to this part of the world. 2015, 2017, when CRISPR here made the cut, it actually became the breakthrough technology by science, and Emmanuel Charpentier and Jennifer Doudna got a Nobel Prize in 2020 for this excellent work they did in uncovering the genetic editing technology that, again, is impacting sickle cell disease. 2018 is when Sickle Gen Africa was launched here in Accra, with our collaborators from Tanzania and Nigeria joining us here in Accra. That same year, our very own Charles Rotimi and his colleagues, using more sophisticated methods than RFLPs, discovered that, in fact, the sickle cell mutation may have occurred in a single person, and thus a more sort of a unicentric origin rather than a multicentric origin that we have studied. So I'm using the strand of DNA as a timeline, and we can still talk about how we're going to unwind this to learn more in the future. But in the next 20 minutes, what I would do is pick key moments, key landmarks that I've shown here, a few of them, to expand on how it has really informed our focus on specific areas of sickle cell genetics and how it will inform us moving forward. For those of you who are not familiar with sickle cell disease, I think it's important to run through some very basic molecular genetics. What you see is a karyotype. The genetic lesion is on the short arm of chromosome 11, where the beta globin gene cluster is located. This is the locus control region, which essentially regulates the efficient expression of all the globin genes in a developmental fashion. The mutation that causes sickle cell disease is a simple A to T mutation in position 6 of the beta globin gene. And this leads to the coding of valine instead of glutamic acid in the beta globin polypeptide. That single amino acid substitution is sufficient to change the biophysical properties of hemoglobin S to the extent that when it's deoxygenated, it polymerizes, causes damage to sickle erythrocytes shown here, causing some of them to become sickled. The sickle hemoglobin sticks to the endothelium, it sticks to other cells. This is the cartoon that Paul Frenet has really given us to help us explain the primary pathophysiology of sickle cell disease. So the number one cellular abnormality of sickle cell is adhesion of erythrocytes. The second cellular abnormality is that the sickle erythrocyte breaks up very easily, releasing cell-free hemoglobin and heme into the vasculature that causes a lot of organ complications. So these two basic cellular anomalies, adhesion, vaso-occlusion, and hemolysis, contribute to a spectrum of clinical complications that impact virtually every organ system in our body. This leads to severe morbidity and high rate of mortality. So sickle cell patients, here this is data from the United States, the median age of survival for men and women is about 20 years lower than their counterparts. This bottom right column is showing you the impact of fetal hemoglobin. This is a study done by Platt, showing that those with high hemoglobin F have a better survival than those that don't. So this is the basic understanding of sickle cell disease. Over the last 45 years, the focus of the genetics of sickle cell disease really has been on understanding how hemoglobin F is switched off and hemoglobin S is switched on. Now that study actually goes further back than 78. This is a picture of Janet Watson. Janet Watson is a pediatrician who worked in New York, and she discovered that babies she was taking care of, who later on developed severe complications of sickle cell disease, when they were babies, when she was looking after them, they were not sick, and their red cells were not sickling as much. So this is the investigator, physician scientist, who made the very first connection between fetal hemoglobin and sickle cell. And this then led to efforts to understand the fetal, the gamma, to the beta switch that essentially has dominated sickle cell disease research over these years. So Janet Watson gets a lot of credit for making that clinical observation, the link between fetal hemoglobin and clinical expression of sickle cell disease. The very first hemoglobin switching meeting took place in 1978. These were powerhouses Athanasios and George Stamatiopoulos, who actually started work and became, I believe, the first president of the Gene Therapy Society in the United States. I'm showing you here a picture of the globin switching meeting in Oxford in 2014. And I'm showing all of this to highlight the focus over several decades on understanding the mechanisms, the genetics of why hemoglobin F goes down and hemoglobin X or hemoglobin A comes up. Just let me let a little bit of advert here. The next hemoglobin switching meeting is going to take place in Crete in Greece in May of next year. And one of the organizers is Doug Engel, and Doug Engel is very interested. Anyone who has interesting work yet to be published in the globin field who wants to participate in the meeting in Greece, if you just shoot me an email, my email's at the bottom there. I'll just forward your details to Doug, and you can join the group to push forward this frontier of understanding of how hemoglobin F is regulated. The extensive work on BCL11A that Stuart 18 and his group did, Stuart Orkin and his group picked up the baton, and here was a publication in Science showing correction of the sickle cell disease in adult mice by interfering with basically fetal hemoglobin silencing. So BCL11A is a potent inhibitor of gamma globin. It silences gamma globin. And if you basically get rid of it in an erythroid-specific fashion, you can correct sickle cell disease. This is a very nice study. This is a transgenic mouse model. So on the left, you're seeing the blood smear of a transgenic mouse that is making human hemoglobin A. This is a litter mate that is making exclusively human hemoglobin S, and you clearly see has the characteristic sickle cells. And here is BCL11A null mouse, and as you can see just from the pictures, that the sickle cell phenotype, at least in the erythrocyte, completely gone. Building on this work, Stuart Orkin gave a very interesting talk, again, with a future word in here. This is the future of human experimental genetics. This was, I believe, organized by Jackson in 2019, where Stuart talked about harnessing genetics to cure sickle cell disease. This has led to a publication this year of successful treatment, at least hematologically, of 6 patients who have sickle cell disease by giving them a lentivirus that essentially inhibits BCL11A. This was published in New England earlier this year. Just to show you some data at baseline, this is the F cells. F cells are erythrocytes that contain a certain amount of hemoglobin F, not 100%. And so for these 6 individuals who went through this gene therapy protocol, at baseline, you can see the percentage of their erythrocytes that contain hemoglobin F, and then following transduction with this lentivirus that inhibits BCL11 silencing, you can see that the F cell, at least for one, shot nearly to 100%. The follow-up, I believe, is actually now 30 months because this was published in January. 6 patients, and the data on the right is showing the hemoglobin F per F cell consistent with this. So this was very exciting. This study started, I believe, in 2020, late 2029. But we had a little slight hiccup when another study using a similar retroviral packaging, a few patients that developed complications to do with, I believe, leukemia, and that led that study to be stopped, and the NIH also paused this particular study. But I know that the FDA in the United States has actually lifted the pause on the other study. So hopefully the issues with the lentiviral has been sorted out, and we can get back on track doing this. CRISPR, as I mentioned, really is the premier gene editing technology, and there have been a study. This was also published earlier this year in January in New England. In this instance, 2 patients with thalassemia, one with thalassemia, one with sickle thalassemia. Just to show you the data, patient 1, if we look at the baseline proportion of hemoglobin, we can tell that this individual has beta thalassemia because they make exclusively really hemoglobin A, but the total hemoglobin is about 9 grams per deciliter, so taken enough. Following infusion with CTX001, this is also targeting BCL11A, by the way, by basically knocking it down, so it does not inhibit beta gamma globin expression. And you can see in this individual, after 18 months, their total hemoglobin is basically 14. I mean, the anemia has been completely corrected. This patient has sickle beta plus thalassemia, but maybe 12% of hemoglobin A, 9% hemoglobin F, and then after transduction. So both of these scenarios are using myeloablative gene therapy. I'll show you a slide as to how that is done in a second. But clearly, using CRISPR and targeting BCL11A, we now have clear evidence that you can genetically cure sickle cell disease. This is a review in JCI asking, are genetic approaches still needed to cure sickle cell disease? And the answer clearly is yes. The 2 papers I shared with you both use myeloablative gene therapy, where hematopoietic stem cells are harvested from patients, ex vivo manipulation with a retroviral vector. In both instances, really targeting BCL11A to prevent the downregulation of gamma globin. Again, the benefits of all those years of doing the hemoglobin switching studies. So we have myeloablative gene therapy, then the gene editing, which the CASPA, which is the second study I showed you. I'm not familiar with any non-myeloablative gene editing, but this is where we want to go because the myeloablation sometimes can be a problem. So this is great. But in 2018, the Independent, this was published in London, said gene therapy is now available, but could cost millions over a lifetime, says scientists. Now, earlier this year, a group did this analysis, which was published in JAMA, and it's quite staggering. The cost of doing gene therapy, the way the 2 studies that I described, is actually over $1 million. And so, whereas the technology is there to do this, it's very, very expensive. And this takes me to this slide. What I'm showing now is a cartoon of the prevalence of sickle cell disease in the world. And as you can see, the majority of cases are in Africa and India. This is a projection for 2010. The projection for 2050 is essentially the same. 2050, I think Nigeria becomes the 3rd or the 5th largest country in the world. And so this is where the sickle cell is a major, major problem. And I just showed you the cost of curing the disease using the current technology we have. Now I want to show you the gross domestic product wealth of the world. This is, how rich countries are. And you can see Africa you can barely see Africa, right? And so there isn't a lot of money in the parts of the world where the disease is most prevalent. Just to show that to you here. So this is the GDP for Ghana, okay, in 2019. It's around $67 billion. Tanzania, $63 billion. Senegal, $24 billion. This is not a lot of money compared to the GDP in the United States, where it is in the trillion, and a trillion is a billion. And so the major problem now is the curative therapies that we have work, but they're very, very expensive. The WHO recognized the importance of developing other means to combat sickle cell disease when it released this paper in 2006. And it said, concerned at the impact of genetic diseases and sickle cell anemia in particular on global mortality, it offered member countries a way to try and control these diseases by proposing 8 recommendations for governments of member states to develop public health approaches that can be potentially less costly and more affordable. Develop medical genetic services, promote relevant, including health counseling, effective international cooperation in combating sickle cell disease, and so on and so forth. To the best of my knowledge, not a single African country has adopted this WHO plan. So whereas the disease is more prevalent here and the cures are astronomical in cost, some of the basic things we can do are really not being done. A major problem is a lot of individual sickle cell disease are not even diagnosed. So when we speak of the genetics, the future genetics of sickle cell disease, even the study that I shared with you, where Khan and Dozie use RFLP to diagnose sickle cell, is not widely available in this part of the world. We did a screen, basically a health screen following this chief when he was celebrating his 20th anniversary, 2019, and we screened 418 children. And we discovered 13 of them had sickle cell disease and did not even know they had sickle cell disease, giving us a prevalence of 3.1%. So undiagnosis is a major problem. The other major problem is misinformation and misdiagnosis. This data I'm showing you is of adults who have voluntarily participated in our Sickle Gen Africa study. This is the type of hemoglobin they reported, self-reported in our case report form. You can see there are only 3 types of hemoglobin states. They said they were either SS, SC, or S-beta thal. But when we analyze the blood samples from this same group of adults, as you can see, we found several other types of hemoglobin. So in fact, 2.5% of these study participants who have been attending the sickle cell clinic didn't really have the right information about that diagnosis. In fact, one of them actually was hemoglobin AA and was attending the clinic. So there's misinformation, misdiagnosis, which is really a major issue. So moving forward, one of the things that we can do is to improve the communication of genetic health information to families, to the general public. We've started to do this. This is a genetic education and counseling for sickle cell conditions, a very short course that we held here a couple of months ago. The course was accredited by the Ghana Psychology Council for 20 continuous professional development points. Because of the nature of the course, we couldn't take a lot of people, but this is the very first time we're doing this, so that the information about genetics, just passing on the information, that people will have the competencies to actually do this well. We're very grateful for the international faculty that participated in this. We had clinical psychologists, genetic counselors. We are very, very grateful to our colleagues in South Africa from Witwatersrand University, University of Cape Town, where genetic counseling programs are actually running. They joined us through Zoom medical geneticist Ambrose Wonka. Some of you are actually listening to this, and I would like to take this opportunity once again to thank you for helping us to essentially do this course and improve the way that genetics is talked about by healthcare professionals. When we ask these participants whether or not the short course met their objective, essentially they all said very good or excellent. When we asked how they will rate the short course, the various components, the course content, the exercise, the role plays, the presentations, all the feedback was good to excellent. These folks graduated, obviously in the COVID era, we're all covering our faces. And here are these guys with their certificates. It was only a 5-day course, so these are not genetic counselors. And we made it very clear to them that having done this short course will not make them genetic counselors, but at least it would improve the way they talk about genetics, the patterns of inheritance. All of them are working with sickle cell families, but they really don't have the right training. What we're trying to do next is to introduce a Master of Science in Genetic Counseling, which will start the early next year. This will be a 2-year course followed by a 1-year internship to train genetic counselors for the first time in sub-Saharan Africa. As I said, there are only 2 programs on the continent, both of them in South Africa. So improving communication of genetic health information is critical. The other critical part in terms of genetics is to understand the genes that modify the disease. Sickle cell disease genomics network of Africa is aimed at doing exactly that. Our goal is to understand how genetics influence the progression of sickle cell disease to inform management and treatment strategies. As much as we support the curative therapeutics, we have to also understand why some people are at risk for some complications and others are not. The SickleGen project is really based, not exclusively, but driven by our understanding of the role of hemolysis in organ dysfunction. This is a review I did with my boss, Mike Gladwin, published in Blood. And essentially our understanding is that when red cells hemolyze and release cell-free hemoglobin, haptoglobin can bind it, but sickle cell patients really don't have a lot of haptoglobin. When hemoglobin is free in circulation, becomes oxidized, it can cause endothelial dysfunction, kidney injury, pulmonary hypertension. Excess cell-free hemoglobin leads to oxidation of the hemoglobin. The heme in hemoglobin becomes free. Free heme and iron can cause oxidative tissue injury, multi-organ damage. We have these proteins that try to block the deleterious effects of cell-free hemoglobin, heme, and iron. And this is really the basis of our work in SickleGen Africa. The idea being that extracellular heme, through signaling through TLR4, causes acute organ damage. We published this in JCI in a mouse model, but the model parallels very closely to the rapidly progressive ACS that has been seen in patients. Recently, studying both patients and mice, we demonstrated that excess heme can also cause acute kidney injury in sickle cell disease. Because whereas excess heme in individuals who don't have sickle cell disease, excess heme is picked up by hemopexin, taken to the liver. This is what is supposed to happen. In sickle cell disease, hemopexin is nearly completely gone. So excess heme is picked up instead by alpha-1 microglobulin, taken to the kidney and causing acute kidney injury. So we know these things can be blocked by these hemolysis defense mechanisms. I've already mentioned the proteins that can do this. And so we basically came together as a group in SickleGen Africa to try and study a large cohort of patients to understand the genomics that influence the levels of these cytoprotective proteins. In Ghana, the project is primarily a GWAS on that. For those 5 proteins, we are now at the stage where we'll start genotyping using the H2 Africa array. And the second project is a malaria project which we've had to modify because really we're not seeing a lot of sickle cell patients reporting to the clinics with malaria. So we're now focusing on an ex vivo project. I will share a couple of slides on the third project, which is a genomic, an echocardiovascular project, where we are again going to interrogate the SNPs that we identify in Project 1 to see whether they will inform us about the echocardiovascular dysfunction that we see in sickle cell disease. This is the organizational, so this is basically the study design for Sickle Gen Africa. We are around here. The project started in 2018, as I showed on that first slide. Our target of 7,000 patients, we've literally recruited all of them. I think we're about 50 shy of that. And there's a lot here. I'm not going to go through all of that in the interest of time, but I just want to focus on the 3 research projects, particularly Project 3, where we are trying to understand how genetics makes one sickle cell patient at risk of developing pulmonary hypertension and another patient not developing pulmonary hypertension. I just want to, again, emphasize genetic counseling when it comes to giving results back. There will have to be some counseling done so that we just don't hand results back to patients without prior counseling. We're already doing this for some of the data, such as the hemoglobin type. As I showed you, some of our participants don't actually have the right information about the type of sickle cell they have. And so for that, we are giving the results back to them. But once we begin to get genotyping data, that we will not do. If there's a need to do that, it will be done with prior counseling. So just one slide on the echo data. This is very exciting. So this is preliminary data. I believe some of you may have seen this. Kofi Ani, who is a collaborator on this project, presented this in the H3 Africa Forum a couple of days ago. So it's very exciting. I believe this is actually the largest cohort of echo data we have in sickle cell disease, period. The previous study, the Walk Fast study, which was done in the US, I think the number was only about 300 patients. And so you can see the mean age. I really want to talk about the TI-jet velocity here. And this is 4.2% of individuals who are TI-jet velocity of greater than 3. This is a high risk for pulmonary hypertension and also mortality. And so moving forward, we are building a cohort that will allow us to interrogate this phenotype and determine whether or not there are specific genetic signatures that predispose a small proportion of patients to develop PH in sickle cell disease. So let me summarize. I don't know how quickly we're doing on time. If we go back to where I started from, sequencing the gene in 1977 till 2008, now we are in a position to study elements of this in Africa. We're doing this with CRISPR technology, editing the basically BCL11A to stop it from being expressed and hemoglobin F not to be silenced. We're doing this with 6 patients haven't really been hematologically cured of sickle cell disease. So how do we move forward? As we make the turn from here on, we are very hopeful of attempts by Gates Foundation and now with Novartis, recognizing that the cost of the the gene therapeutic products we have, basically it's not feasible. $1 million is a lot of money. And so as we move forward, the expectation is that there'll be cheaper and more accessible gene-based therapeutics that can be afforded by the majority of patients in places like Ghana, where I'm talking to you from, Tanzania, and also parts of India. Very often we don't remember to talk about India, but India has a very large population of sickle cell patients. Brazil, all these places where the disease is prevalent, the current price tag for curing the disease is really beyond them. Moving forward, this will be great, that one will be able to couple newborn screening, which is still a challenge in this part of the world, with autologous therapy. Once the baby has been diagnosed with sickle cell disease, again, the information, how the information is relayed is very important. So genetic counseling is going to be very important. So we need a comprehensive approach of prenatal counseling. If a child is at risk or pregnancy is at risk of sickle cell disease, competency in relaying that information. We also, moving forward, some of these ex vivo manipulation. Clearly, laboratories in Africa, India, Brazil will have to be equipped to do this. If we're going to do autologous transplant, somehow we have to have laboratories here that can, at least hematologists as well, that can actually recover hematopoietic stem cells, mobilize these cells, do the ex vivo correction, and then pass it back into babies who may have sickle cell disease. Whilst this is great, there are millions of adults who already have sickle cell disease. So how do we move forward? This is my last slide. And moving forward, it's going to be very important to incorporate data science to improve the phenotyping of sickle cell disease. So deep phenotyping is going to be critical for us to integrate deep phenotyping with the genetics. And all of this has to be done with cross-cutting themes that are focused on these three principles, I believe. Community and public engagement regarding new diagnostic and curative therapies. The community has to be engaged. The community has to be told what is happening. Training at-risk populations in Africa and India in all aspects of genetic medicine. And then developing, I can't see the last part because of this. I guess it's cheap and affordable therapeutics, whether it's genomics, epigenetics, and pharmacology. I want to thank you. I'm speaking to you from one of the offices, an office right here, the West African Genetic Medicine Center. Once again, I want to thank the organizers for inviting me to share my perspective on the way moving forward. And what I'm saying is we'll do that by looking back and learning from what we've done in the past to help us move forward. Thank you very much.
Thank you so much, Solomon. That was really an incredible journey, taking us, from the basic principles and right catapulting us into the present and also thinking about the future. I think it's extraordinary how much has happened in terms of therapeutics, even in the last 2 years. I think, for me, listening to you and covering all the bases that, I often wonder whether we need to go back to the basis of
Oops. Oh dear. Can you hear me? Can you hear?
Yes. Okay. Can you? Okay. Thank you, Solomon, for a very great presentation. And from questions and answers session, let me see. Oops. I see nothing. Yes. Yes, I'm there. Yeah. Okay. We have a couple of questions and comments from Julie and Khalid. From Julie, I would like to recognize and thank Salomon for in his story of sickle cell disease in Africa. His leadership and support was instrumental in the process of sickle in Africa. Without Solomon sharing his experience and expertise in NIH grant application, the Spark grant would not have been successful. Thank you, Solomon. The HPC has been extremely valuable for our research. This is mostly a comment. I don't know if you have any.
Thank you, Julie. Thank you, Julie. Wish you well.
And I think from Khalid, is it from? It's just an opinion. How can African government deal with the problem of high cost of curative treatment of sickle cell disease? The most relevant studies are done on African patients, and when we found a curative treatment, African counter could not take it in charge the patient. So the Nobel Prize of the research is somehow not really reached.
Yeah, so I think we have to continue to contribute as best as we can to the development and refinement of these therapeutics so that they become affordable, right? So we have to get engaged. They are expensive, but there are ways to try and develop cheaper versions. And I think we have to, this is why I think it's very important that we leverage the work that we're currently doing so we can contribute to the wet bench work, the work in the laboratories, the transductions. If we do that, then we can help reduce the cost as we develop new packaging, viral packages to transduce the cells. The technology is there.
Okay, thank you, Prof. Solomon. There is one more question from I think from Edmond Uncam. He says, "Thank you for the great presentation, Prof. Solomon. Please, why is hemopexin level reduced in ACD patient? And do you think exploring this can help prevent acute kidney injury in ACD patients?"
Yes, so hemopexin is the primary heme scavenger protein. It binds to heme and takes it to the liver to degrade heme. And because of the intensity and the chronicity of hemolysis, sickle cell patients basically run out of hemopexin. And so one simple way, and I know there's a drug company that is currently doing a clinical trial of hemopexin replacement therapy. And so if you replace the hemopexin, just like replace factor VIII for hemophilia, then the bad effects of heme can be mitigated. So there is actually a company trying to do this right now.
Much Gaston for thank you, Solomon. I'm sorry that I disappeared. So just to say, Solomon, lots of compliments. You can have a look in a minute. And really thank you for your leadership in this area and for this absolutely extraordinary talk. And Gaston, I'm going to hand back to you now and we'll leave the stage. I'm sorry that we're now 15 minutes behind schedule. Thank you, everyone. Goodbye.
Yeah,
guys, enjoy the meeting. Bye-bye.
Thank you, Solomon, for a great presentation. So thank you, everyone. We'll be now having 3 presentations in this session from Aimé Lumuka, Heaven Life, Christopher, and Florence Urio. So we'll start with Dr. Aimé Lumuka. presenting on pharmacogenomics of sickle cell disease. So it's a great pleasure for me to introduce to you Dr. Aimé Lumuka, who is a professor of genetics and pediatrics at the University of Kinshasa in Democratic Republic of Congo, and also the coordinator of genomics medicine platform at the University of Liège in Belgium. His current work focuses on the use of whole genome sequencing to foster the identification of novel development genes and improve care in severely ill neonates. Prof. Lumuka is leading the initiative of the development of an African reference database for rare diseases, and he's a chair of rare diseases working group for H3 Africa. His contribution to science include the identification of genetic anomalies causing developmental disease in African patients, description of African phenotype in non-genetic diseases, and identification of genetic modifier of the phenotype and treating response in sickle cell anemia. Professor Lumuka is a medical doctor by training from the University of Kinshasa and obtained his PhD degree in human genetics and genomics at Leuven University in Belgium. He worked as chief medical officer in Belgium-based company deployed, currently part of Invite, where he oversaw clinical interpretation of next generation sequencing data and collaborated in the development of artificial intelligence-based genome interpretation tool. So he's today our presenter and will be presenting on pharmacogenomics of sickle cell disease. So, Professor Aimé Lumuka, the floor is yours. Take it.
Thank you, Gaston. Thank you to the organizer. First of all, I would like to know which of my screen you are seeing. Is it the presenter mode or the normal diapo? Okay, let me just change which screen I'm sharing. Okay, so thank you very much to the organizing committee for inviting me to talk about the work we are doing in the Democratic Republic of Congo on sickle cell anemia. And before I dive into this work that we are doing currently, I wanted to bring us back to where and when the research on sickle cell disease anemia started in DRC. We are the third biggest country in Africa with more than 90 million inhabitants. And in our country, sickle cell anemia account for 2% of the newborns. So we are, from a geographic point of view, well positioned to inform on migration, gene flow, and how could have influenced the adaptation and the population distribution, and also could have an effect on the phenotype and the response to treatment as well. The first phase of research on sickle cell anemia, as far as I was able to retrieve in the literature, the first publication was made in 1952 by a team of researchers from the University of Leuven. This is the mother university that actually was at the origin of the University of Kinshasa. And the work they did from this publication of 1952 to 1966, this is the last publication they put out. It was more about describing the phenotype and also trying to understand how other diseases could affect the phenotype of sickle cell diseases. And the second phase was led by the late Professor Kabakele. He was a surgeon and was specialized in bone diseases. He was a traumatologist. And he came to the field because sickle cell patients, they always have the wounds that are not healing, and those ulcers. He was one of those that developed surgical approaches on how to treat those patients. So he was also at the origin of the biggest sickle cell research center in our country. The third phase, I believe this is the phase we are in now, and one of the pioneers in this third phase is Leo Chilolo, that most of us know very well. He has been conducting researches since 1992, starting by describing the phenotype, and now he has dived into exploring also the genetics, which genetic factors in our setting would influence the phenotype of sickle cell anemia and could also influence the response to the treatment. Although he has not yet published this part of the study, that we know that he is already looking at this type of approaches as well. And then we, the Center for Human Genetics of the University of Kinshasa, we also came into this field very recently, and these are our few publications from the description of the clinical phenotype, the description of the severity of the phenotype in Congolese patients. We have also quickly evolved into studying the genes. This is our core business, looking at the genes and how the genetic determinant of one individual could affect his baseline phenotype. And this publication that we made in 2018 was actually the one that built or established the foundation of the work we are doing now. From the moment that we could replicate that genetic factors are modifying the phenotype in Congolese patients, and from the fact that more and more publications are being put out that are showing the influence of the same genetic factors on the response to hydroxyurea, and additional genetic factors that could also influence genetic response to this well-used treatment, we thought that it would be interesting to go further. In the first study, we explored 8 polymorphisms, and we thought that it was time to increase the number of polymorphisms that we could explore. So we developed another study which remains a candidate gene study, because we are not conducting GWAS, but we are targeting SNPs that has been published in the literature or deposited in GWAS catalog, where there is a strong association that has been established between the gene and either the baseline values of fetal hemoglobin or an association with an effect or the metabolization of the hydroxyurea, and other SNPs that have been associated not with the baseline, but only with the effect of hydroxyurea. So from our literature search, we actually identified 56 SNPs that we thought that would be worth studying in our population. This included 13 SNPs that had been previously associated to the baseline levels of fetal hemoglobin, 28 associated to the effect of hydroxyurea on fetal hemoglobin, and we also had 10 polymorphisms that have been associated with both the baseline value, and the level after the treatment with hydroxyurea. We also included one SNP that has been associated with the liver metabolization of this drug. And in addition, we also wanted to look at the haplotypes of sickle cell anemia in our country, since this is one of the information that were not available yet. We commonly use the imputation from data that has been published in the neighboring countries. We were lacking data from our own population to know which haplotypes are common and how those are distributed. So we took this as an opportunity to go beyond and try to explore the haplotypes in our community. This study was conducted in 2 hospitals. One is the center that I talked about in a couple of slides ago, that was developed by Professor Kabakele, which is the reference center for sickle cell anemia in DR Congo. And the name in French is Centre de Médecine Mixte et d'Anémie SS. And this one is located in Kinshasa, the capital city of Congo. But the study was also conducted in a rural area in the hospital named Saint-Luc Hospital in the province of Congo Central. So we combine the urban and the rural setting for this study. One of the things that is important here in the rural setting, this particular hospital is only looking after pediatric patients. So we excluded pediatric patients from the urban setting so that we could have a sort of representation where we have children from one side and adults from the other side. All the information and samples were coming to the Center for Human Genetics of the University of Kinshasa, where we were able to actually perform hemoglobin typing using capillary electrophoresis and to extract DNA from those patients. We were also able to do the confirmation of the diagnosis through a molecular testing, and Solomon talked about, and the next speaker is also going to talk about. We developed this approach because many of the patients that are labeled as sickle cell anemia, and some of them are even receiving treatments, are actually not sickle cell anemia. They have other problems, but they are not sickle cell anemia. So to be included in this study, we had to conduct this test, to perform the molecular test to confirm that the patient that is included in this study is truly a sickle cell anemia patient. And then DNA was sent to the K11 in Belgium, where PhD students within this project are registered. This is where they are actually doing the PhD thesis. So to be included in this study, patients needed to be part of the cohort of sickle cell anemia in one of the institutions where we were recruiting. The second, we had to validate the sickle cell anemia status using a DNA-based test. It's a resection fragment length polymorphism test with the digestion enzyme that is actually available at the University of Kinshasa. The patient have to have not received a blood transfusion the 3 months preceding the recruitment. Of course, they have to provide a consent signed either by adult patients or by parent when the patient was a child. They had also to be available to provide blood sample, not only for the recruitment, but also for the follow-up that were set in the frame of this study. So after the initial assessment, which included the recruitment, the consent process, and the formal recruitment, the primary clinical examination, and based on the primary values, patient was actually classified into 2 groups based on the level of the fetal hemoglobin. Those with fetal hemoglobin equal or above 15, they only receive the molecular result of the DNA test we performed at the University of Kinshasa, but they could also keep coming to the 3-monthly consultation that our team set in either of those institutions. But for those who have the baseline level of fetal hemoglobin below 15%, those were included in the trial that we set up. So in this trial, the hydroxyurea was provided to patients starting with a dose of 10 to 15 milligram per kilogram. And we used an escalation of 5 milligram level, and this was a monthly escalation until we reached the maximum tolerated dose, which was 30 to 35 milligram per kilogram. Every 3 months, all the patients were seen at a consultation in the institution where they were recruited. During those visits, a clinical evaluation was performed. We were looking at the adverse drug reaction when they have not been reported within the window of 3 months. We actively looked at those adverse drug reactions. We also sampled for blood testing to control for fetal hemoglobin, white blood count, reticulocyte platelet, and LDH. So at the end of the trial, this is the operational definition we use to separate patients between responders and non-responders. This is a definition that we borrowed from this publication, where a respondent was someone who had reached a plateau of fetal hemoglobin above 20%, or someone, even if the plateau was not reached, but someone that tripled the initial level of the fetal hemoglobin. This one also was considered as a respondent. And this is the simplified description of the single molecular inversion probe library preparation, where you have this construct that is designed by the manufacturer based on the data that we provide, the target that we provide to the manufacturer. And this construct includes at either end a target that is specific for the sequences that are flanking your SNP of interest. And then you have a tag that is specific to that particular SNP. You have a primer site that will help to amplify this fragment. And when these fragments is hybridized on DNA, the sequence that are specific to the flanking of the polymorphism that you are exploring, this one is the one that is going to ligate actually to your native DNA. And this will circularize during a PCR process. Non-circularized DNA will be removed by exonuclease. So at the end, you have your fragment that has been amplified. They have been actually tagged with a patient-specific code bar and are multiplexed. This library is going on to the MySeq. This is where our sequencing was performed as a paired end on a flow cell with 200 cycles, 2 times 100 cycles. So what we have observed, this is the general characteristic of the population. Before we go into the clinical trial, our cohort consisted of 275 individuals. So the distribution of genders was actually skewed, as you would see on this graph of the haplotype that we are just showing this, not to discuss the haplotype, but to give you a visual idea of the gender distribution. As patients were older, we observed that there were significantly more female than male in our cohort. The age ranged from 1 to 40 years. At the baseline, at the beginning, when we had the 275, we looked at the baseline values. What we observed out of the 275 is that there was a sort of difference that was very significant for the fetal hemoglobin, the hemoglobin in general, the fetal hemoglobin, the white blood cells, and the reticulocyte and platelets. When we compared Kinshasa to Kisantu, as I said, Kinshasa were adult patients and Kisantu children. So we observed this difference between adults and children. But for some of those parameters, the significance was not confirmed when we performed the Bonferroni correction. For those who were included in the trial, in total 105 were initially included in the trial, but 25 were later excluded, including 2 that were deceased. They were excluded for diverse reasons, and this is not discussed in this presentation that we are giving today. The age ranged from 2 to 38 years. So before the hydroxyurea started, when we zoom into the initial data for the 77 patients that finished the clinical trial, we observed some differences in the value of the fetal hemoglobin between genders and between ages. This value was higher in female and was higher in children. After the clinical trial, when we separated patient into respondent and non-respondent, we observed more respondents than non-respondent in this cohort. And when we compared the proportion of respondent between children and adults, we didn't observe any significant difference for that. That cannot be ascribed to the age of the participants. So here we are presenting the evolution of the fetal hemoglobin over the course of the 24 months of the research. As you would see, we observed a significant increase in the fetal hemoglobin in the group of respondents. This is actually the main criteria that define respondent between and non-respondent. So in a way, this is a kind of circular reasoning because this is the only criteria that is separating the two groups. You can see that there is an evolution, there is an increase within the two groups, but this increase is much more higher in the group of respondents. But when we compared the respondent and non-respondent for the white blood cells, we did not observe any difference that was created for the white blood cells. In both groups, we observed that the hydroxyurea treatment actually decreased the amount of the number of white blood cells, but the decrease was not significantly different between the two groups. The same trend was also observed that we could see that there was a change in the value of reticulocyte, but the difference was not significant. The same also applied for the LDH. And when we look at the data that were generated by sequencing the 56 SNPs that were retrieved through the literature, the first thing that we did is to make sure that the variation that we could see looking at the results of the clinical trial would not reflect any change that would be at the population level. So the first thing that we did, we explored the Hardy-Weinberg equilibrium for each of those SNP at the level of the population, which is the 275. So out of those 56 polymorphisms, 4 were actually not balanced in the population, which means that there was a skewing. They were not fitting with the expected frequencies for those SNPs in the population. So those 4 SNPs were further excluded for the clinical trial evaluation. When we evaluated the remaining 52 SNPs with regard to the response to the hydroxyurea between the respondent and the non-respondent, we did not observe any significant influence of for 48 SNPs, only 4 actually showed a significant correlation or influence on the level of response achieved or the level of fetal hemoglobin that was achieved at the end of the clinical trial. So we have actually, as I have shown through the story of sickle cell anemia in DR Congo, set an environment for genomic research on sickle cell anemia in DRC. And this study illustrates the complexity and the difficulty of running a proper clinical trial in a research-limited setting. We have observed that some of the well-known trends were also present, but the significance was not reached. We don't know whether the sample size could be one of the reasons, but only 4 SNP actually correlated with the level of fetal hemoglobin expected after hydroxyurea treatment. Even some of the correlation we observed in our previous study of 8 SNPs were not significantly confirmed here. So maybe a GWAS is needed to explore Congolese patients to identify much more SNPs that could be associated with both the baseline phenotype and the response to hydroxyurea. We hope that with further collaboration and fundings, we would be able to dive deep into the genotype influence of the baseline phenotype and the hydroxyurea treatment in Congolese patients. So I would like to thank the funders. This research was mainly funded by the Rios, and we also received a strong support from the University of Leuven in Belgium. And this is the team that is driving this research. Thank you very much.
Aimé Lumuka for the great presentation. And we have to wait for question and answer event at the end of all presentations so that we can have nice discussion. So I'm going to pass the floor to Professor Julie to introduce the 2 last presenters. Prof. Julie, the floor is yours.
Thank you. Thank you very much. That was a fantastic talk, Dr. Aimé Lumuka from DRC. Really, really great work that you're doing. So I will now introduce the 2 speakers. Heaven Light will speak first, followed by Florence. So Heaven Light has a Bachelor of Medical Laboratory Science and is currently a Master's Fellow at Muimbili University. She will talk about her work on DNA testing for sickle cell disease. And this is work that she has been doing with Dr. Siana Nkia, who is the president of the Tanzania Society of Human Genetics and a scientist, and Professor Anna Shu, who spoke earlier about diagnostics in leukemia. Heaven Light has also been integral in newborn screening for sickle cell disease, which is part of the CONSA work with the American Society of Hematology, which is conducting newborn screening in more than 6 countries in Africa. And she's also working very closely with Mohammed on looking at hematopoietic stem cells for gene therapy in sickle cell disease in Tanzania, and is very much working with Florence as well as clinicians in exchange blood transfusion for sickle cell disease as a service. The next speaker, after Heaven Light, Florence will speak, and Florence is a fantastic scientist. She has been working very closely with Lucho, looking at malaria and fetal hemoglobin, HbF, as well as sickle hemoglobin. And she and Lucho have set up ex vivo studies where they're looking at the pathogenesis of disease in understanding malaria pathogenesis in individuals with different levels of HBF, HBS, and HBA. As a PhD fellow, she has published more than 6 papers in international journals. She really is a fantastic scientist, and she's working with Siana and investigators in sickle in Africa as part of the first phase of SPARKLE, where they're conducting a multi-country study in Ghana, Nigeria, and Tanzania, looking at HBF in sickle cell patients in these 3 countries. She's also been, as part of her work on malaria, she's also been looking at malaria and sickle cell disease, particularly during the COVID-19 pandemic. And we're hoping that we'll be able to extend this work as part of the work in Sickle Gen Africa or in Sickle in Africa. So she will be talking about the discovery that she has made, which is really a novel discovery of an X-linked factor that is associated with fetal hemoglobin and particularly F cells. And this is really a novel discovery that has been made and was published this year in BJ Him. So with those 2 introductions, I'd like really to welcome Heaven Light from Muimbili University, the Sickle Cell Program. Welcome, Heaven Light.
Hello everyone, my name is Heaven Light Christopher. I'm a laboratory scientist at Muimbili University of Health and Allied Sciences and the Department of Hematology and Blood Transfusion. I will present to you my research project titled Using DNA Testing for Precise, Definite and Low-Cost Diagnosis of Sickle Cell Disease and Other Hemoglobinopathies. I will take you through the background, objectives, methods, result, conclusion, and finally the acknowledgement. Globally, it is estimated that 300,000 babies are born with sickle cell disease annually, with majority of this baby being born in sub-Saharan Africa, and it's where the mortality of babies before the age of 5 is estimated to be as high as 50 to 8%. And Tanzania is the 5th country in the world with the highest sickle cell disease birth prevalence, with an estimation of baby being born with sickle cell disease to be 11,000 annually, and without the proper diagnosis and management, 50% or more of this baby may die before the age of 18, with the highest mortality being under the age of 5 years old. To reduce the burden of sickle cell disease, there were various initiatives that were introduced to reduce the mortality and morbidity that occur due to the sickle cell disease, and of those initiative, one was establishment of newborn screening for sickle cell disease with the aim of screening newborn at birth, and the newborn who were found to have sickle cell disease were immediately enrolled in a comprehensive care, and it's where the management and monitoring of these patients were followed up closely in order to reduce the pain crisis and also to improve the quality of life of patients with sickle cell disease. But also there were introduction of education to parents and caregivers who are responsible for taking care of children with sickle cell disease, with the aim of improving the awareness and knowledge regarding sickle cell disease, the manifestation of sickle cell disease, but also on how to manage and take care of the children who have sickle cell disease. But also there were community engagement and awareness regarding the sickle cell disease to provide knowledge to community regarding the disease itself and how the disease is inherited, and to remove the myths that were that occur due to sickle cell disease. On my right hand side, the first baby that was diagnosed with sickle cell disease in Tanzania in 2015 at Temeke Referral Hospital through the newborn screen service that are currently ongoing. Despite the various initiatives that were put in place to reduce the burden of sickle cell disease, unfortunately no country in Africa has successfully established newborn screening as a universal national health intervention. This is mainly due to the cost and complex logistics of the currently available services, and also the diagnosis of sickle cell disease in certain cases, such as sickle cell disease that occur together with thalassemias, requires DNA analysis, which is currently very expensive. In order to address one of the challenges that faces or hinders the establishment of newborn screening as a universal national health intervention, we thought worked on developing a precision mobile DNA technology that will be able to diagnose the sickle cell disease and other hemoglobinopathies, with a specific aim of testing the performance of the assay on DNA from both whole blood sample and dried blood spot samples, but also to compare the nanopore ion analysis test with the conventional screening diagnostic method, that is isolytic focusing, high performance liquid chromatograph, and Sanger sequencing. We conducted our study at 2 hospitals. One was Temeke Referral Hospital, and this hospital is currently conducting a newborn screening for sickle cell disease and other hemoglobinopathies. And Muhimbili National Hospital is actually the hospitals that conduct sickle cell disease clinic every weekly in Tanzania. And our study design was retrospective and cross-section. Our study populations involved 18 newborn that were delivered at Temeke Referral Hospital and 18 adults who attend a sickle cell disease clinic at Muhimbili National Hospital. And we use the convenient sampling as our recruitment method. We grouped our sample into 2 cohorts, the validation cohort and discovery cohort. In validation cohort, we involved 15 samples. 5 were from dried blood spot that come from newborns, and 6 were whole blood samples that come from adults who attend sickle cell disease clinic. And this sample from validation cohort were previous sequenced by Sanger sequencing in order to confirm the beta thalassemia, and also to confirm some of claim of sickle cell disease carrier patient who claim to have symptom of patient who has actually sickle cell disease. In discovery cohort, we involved 21 samples. 9 were dried blood spot samples from newborn, and 12 were whole blood sample from adult. And this sample were not previous sequenced by NL sequencing method. In this research, we use the Oxford nanopore technology as our precision mobile technology of choice. And the Oxford nanopore technology is a sequencing technology that enables the direct real-time analysis of a long DNA by monitoring the changes to an electrical current as a nucleic acid passes through the protein nanopore of the flow cell. In sequencing and analysis, as I presented before, our samples were previously screened for Sanger sequencing, especially in validation cohort, and for both samples in validation cohort and discovery cohort were processed for full body picture and isolytic focusing and HPLC, and their results were recorded in our database. And after that, from whole blood sample, we separated the Baficot and stored it at -80 for further analysis. And during the DNA extraction, we extracted our DNA from both Baficot and dried blood spot by using the Kayam DNA blood min kit, and the DNA concentration was quantified by using the Qubit 3.0 fluorimeter, and we use both assays, broad range assays and high sensitivity assays for measuring our DNA concentration. And after quantifying our DNA, then we proceed to amplifying our target gene, which is beta globin gene locus, and this was done by using the modified primer with attached ONT per-coding tail sequences. And after that, we did the purification of our DNA by using the Ampure XP beads, and the concentration of our target gene was measured and recorded. We pulled our DNA to a desired ratio for sequencing, and after that we ligated and prepped our DNA by using the ligation sequencing kit protocol. In here we basically attach the specific adapter that will hold the DNA long enough in a flow cell for its nucleotide to be identified electronically in a real-time manner. The sequencing was performed by using the flow MinION 107 flow cell on an instrument called MinION MK1B for 2 hours, and we were able to generate more than 0.4 gigabyte of data with a quality score of 20 and a quality depth of more than 36,000. And after the sequencing, then our raw sequenced data was base coded and multiplexed by using the copy software, and the alignment was done by using Minimap2 software. The variant calling was done by using VarScan, and the annotation was done by using the ANNOVAR. These are basically the analysis steps that were followed during the bioinformatics of our sample. The result of objective 1, that was to test the performance of assay on DNA from both whole blood and dead blood spot. We were able to identify the mutation on a beta gene locus on both whole blood spot sample and the dried blood spot samples. A total of 75 germline mutations were identified, 38 were synonymous mutation, 25 were non-synonymous mutations, 4 were a loss of HBB initiation codon, and 2 were splice mutation located at exon 1, and in the bracket it shows the specific mutations that were identified at exon 1, and 6 were stop gain mutations that were located at exon 2, and in the bracket it shows exactly what kind of mutations that were identified at exon 2. And the result for object 2, that was to compare the result of the MinION DNA analysis test versus those of conventional screening methods, that is IF, HPLC, and Sanger sequencing. And from this table, from my left hand side, it shows the sample that were included in your study, the sample type, the cohort, both validation and discovery cohort were included for this objective, and the methods that were used in conventional screening methods. So some of the sample we use Sanger sequencing and HPLC. A sample for dried blood spot, the conventional screening method that was used was selective focusing, and sample for whole blood, the conventional screening method that was used was Sanger sequencing. And the result from conventional screening method is as shown in the table. So as you can see from both conventional screening method and the result from the nanopore screening method are exactly the same, with a few exceptions in few samples. As you can see in sample 144 and 245, in nanopore we were able to detect 2 different mutations in the same gene locus. As you can see in sample 245, for example, we're able to detect the triple FAT-X mutation, but also we're able to detect the root 7 value mutations that cause sickle cell disease, and triple FAT-X mutations that cause the thalassemia mutation. And this table is an extension of all the samples that were performed for the objective number 2, that was to compare the result from conventional screening method and the result from nanopore sequencing method. To conclude, our sequencing result for sickle cell disease uses the MinION instrument sequencing method were 100% same as those from Sanger sequencing method and other conventional screening methods, that is high performance liquid chromatograph and isolytic focusing. In addition, the long link DNA sequencing method enabled the resolution of cases with unusual phenotype, which make up 1% for all children in Africa. But also the turnaround time of MinION sequencing technology is actually shorter as compared to the conventional sequencing method. As explained before, we were able to sequence a sample for only 2 hours, while other conventional sequencing method may take more than 24 hours. But also, as explained before, our instrument is very simple, easy to use, and you can use it anywhere. It doesn't need a comprehensive building for sequencing to be done. As long as you have computers that have charge, then you can use your instrument everywhere. So this, we thought it will be a very good sequencing technology, especially in limited resource countries. And finally, I would like to acknowledge the sickle cell disease patient and their families who gave us the permission to use their samples for this research. But also I would like to thank the staff of Department of Hematology and Blood Transfusion Sickle Cell Program, but also the staff from Temeke Referral Hospital and Muhimbili National Hospital. But also I would like to extend my gratitude to the funders who funded this research, the Organization for Women in Science for Developing World, the UK Medical Research Fund, and the Wellcome Trust Oxford Translation Fund. Thank you for listening.
Greetings everyone, my name is Florence Urio. I'm a PhD fellow from Muhimbili Sickle Cell Program, but I'm also an assistant lecturer from Muhimbili University of Health and Allied Sciences. I'm going to give a talk on association between genetic variants and F cells in sickle cell disease. Tanzania is populated with over 58 million people, of which children who are born with sickle cell disease per year are estimated to be 11,000 babies, and deaths due to sickle cell disease of below 5 years are estimated to be around 1,500,000. And currently in Tanzania, there's only one drug that is available for treatment of sickle cell disease. Hence, there is a need for research to understand the mechanisms of disease in order to develop new interventions. So sickle cell disease was actually the first human monogenic disorder to be characterized at the molecular level. And this is just a schematic diagram that illustrates how the point mutation occurs in sickle cell disease. So at the gene of the beta globin gene, at the 6th position where you have adenine, there is a point mutation at this position. Instead of having adenine, you end up with thymine, and as a result, instead of having glutamic acid amino acid, you end up with the valine amino acid. This produces a mutant globin protein, and the glutamic acid is actually an hydrophilic amino acid, and valine is an hydrophobic amino acid. So the abnormal hemoglobin in which valine has replaced glutamic acid causes the hemoglobin to be less soluble under hypoxic condition, and therefore this hemoglobin they do polymerize into crystals that distort the red blood cells into sickle shape. As you can see here, these patients tend to have these sickle shapes. They tend to have these sickle cells, and these sickle cells do block the blood flow in the blood vessels, and as a result, these patients tend to have pain in the joints, and actually this is one of the pathophysiology of the sickle cell disease. So fetal hemoglobin, which is the predominant hemoglobin at the gestation age of 40 up to the time when the babies are 2 years, is one of the major modifiers of sickle cell disease severity. And this is just in an illustration that shows the erythropoiesis process that takes place in the normal individual and in the sickle cell disease individual on the right. So on the left, this is the marrow of a normal individual, whereby there is a gene switch from gamma to beta, and as we know that the normal adults normally have higher percentages of adult hemoglobin variant as compared to other hemoglobin variants. So during the gene switch, the switching is not so complete in such a way that we tend to retain some small amount of fetal hemoglobin within our red cells, and this fetal hemoglobin is retained in a subset of cells known as F cells. So a normal individual has below 1% of fetal hemoglobin and below 7% of F cells, and majority of the hemoglobin is the adult hemoglobin. Whereas in sickle cell disease individual, we know that these individuals do undergo hemolysis. So in order for the bone marrow to compensate for the loss of these red blood cells, it undergoes stress erythropoiesis, and as a result, there's accelerated erythropoiesis, and there's an increased levels of fetal hemoglobin being produced, and subsequently higher levels of F cells will be present in these individuals with sickle cell disease. So the levels of fetal hemoglobin and HbF is not stable within in these individuals. There's a high variation amongst these individuals with sickle cell disease. There are many factors that influence fetal hemoglobin and F cell levels that influences this variation that is seen in sickle cell disease individuals. And some of these factors include age, sex, the alpha globin gene number, the X-linked locus, as well as preferential survival of F cells in peripheral blood. And the number of F cells in sickle cell disease patients actually depends on two main factors. One is the increase in proportion of erythroid cells that still make the gamma chains as a result of stress erythropoiesis, as we've seen in the previous slide, but also the preferential survival of F cells in the peripheral blood because of their relative resistance to sickling, as we know that these F cells do not undergo the polymerization process due to the tetramer structure of the fetal hemoglobin. So as we've mentioned, there are many factors that influence the variation that is seen in sickle cell disease individuals. Genetic factors are one of the major factors that influence this variation. And previous studies have looked into these genetic factors, and they've identified 3 known loci in chromosome 2, 6, and 11. However, these loci only explain about 20% of the variation of these fetal hemoglobin and F cells that are seen in most of the sickle cell disease patients of African ancestry. Henceforth, there is a need for further studies to investigate this missing heritability of fetal hemoglobin and F cells in sickle cell disease. And hence, in our study, we aimed to investigate how F cell percentages rather than fetal hemoglobin correlate with their polymorphic genetic determinants. Our study was actually carried out at the sickle cell disease clinics at Mwananyamala Temeke na Mana in Dar es Salaam, Tanzania. And this included, our study design was retrospective and cross-sectional study. And our study population were actually sickle cell disease individuals that were attending the sickle cell disease clinics. We used convenience sampling to select for the study participants. In order to do the statistical analysis, we used Stata version 14 as well as the RStudio. For descriptive statistics, we used median and interquartile ranges for the continuous variables. And in order to test for gender differences, we used the Wilcoxon rank sum test. To determine the effect of different genetic variants on F cells and HbF, we used the univariate regression model. So this is just a flowchart that illustrates how the sample size was generated. So my colleague and mentor, Dr. Siana, had generated data of about 2,000 individuals. So she conducted a study, actually it was one of the largest genome-wide association study of sickle cell disease individuals in Africa. And so we have data of these about 2,000 individuals, of which out of these, 1,400 had HbF level measurements. From this 1,400, those that were above, those that were 5 years and above were about 1,200. And from this, we were able to recall by using the convenience sampling, and we ended up with the 227 patients. However, 5 of these individuals did have contradicting HbF levels, and therefore we ended up with a sample size which was 222. So as I said, we wanted to evaluate the association between genetic variants and F cells. First, enumerate the F cells, and we use the Fax caliber machine. As you can see in this picture, this is a machine that we have in our university, and it's a 4-color machine. And before acquiring the data, we had to first fix the cells using the glutaraldehyde, and then we had to permeabilize using Triton X, and finally stain with the anti-HbF. After this, we acquired the data in the machine, and we did the analysis using the FlowJo software. And this are just the results from the FlowJo, where the results can be presented either in the dot plot or in the histogram format. and for this particular individual has about 42.3% of the F cells. And looking at our findings, we did look at the gender differences in hematological parameters of the study participants, and we observed a statistically significant difference between males and females in all of the red cell indices except for hemoglobin and MCHC, but we also found there was no statistically significant difference in the white blood cells count as well as the platelets count. We also observed a different distribution of F cell percentages in females compared to males, and this was actually similar to a study by Creel et al. In 2009. And this fact actually led us to hypothesize that some or most of the differences that we've seen in the previous table may be a consequence of gender difference in F cells percentages. So we went further to investigate the X-linked XP22.2, which is the F cell production locus, which was previously studied. So the conspicuous difference in F cell percentages distribution between males and females was actually suggestive of an X-linked genetic determinant. So this locus was actually the first studied by Dover et al. In 1992 through a pedigree analysis in some families with sickle cell disease, and this led them to postulate that an X chromosomal dominant genetic determinant might be on the XP22.2. So we tested our variable, which was fetal hemoglobin and F cells, and we treated this with as continuous variable, and we used a candidate SNP approach. We picked the two candidate SNPs from studies by Soloviev et al. In 2010 and Chang et al. In 1995, and these two SNPs reside in the FRMPD4 candidate gene. And when we did this association, we observed when we did this test, we observed that there was no association. We then tested the association of F cells separately between males and females using arbitrary cutoff values in F cells of 20% in males and 40% in females. We found a significant association with the RS16998911 that is found at this region within the 5' untranslated region of FRMPD4 in males with high F cells. So since 20% cutoff value was arbitrary, we went further to explore different cutoff values both in females and males, and we can clearly see that in females across all the F cell percentages, we see that there is no significant association. However, in males, we were able to see an association to be robust over a range from 13% of F cells up to 30% of F cells. We also scanned the chromosomal region of XP22.2, which comprises of about over 4,000 SNPs, and we observed that 3 SNPs in males and 1 SNP in females were above P value of 10 to the power of -4 bar, which tagged the FRMPD4, and adjacent to this was the MSL3 and the TL8 genes. However, a signal that reached the threshold of GWAS significance, which is 10 to the power of -6, was confirmed in males for HbF percentages against the RS16998911, and this was actually the only signal amongst the 4 stated in the previously to have reached the criteria of significance. So apart from testing the FRMPD4 gene with the candidate SNPs, we were also interested to look at whether the previously known loci were associating with variables from our population, and we found that from the 3 on chromosome 2, 6, and 11, we found that there was a statistically significant association in BCL11A, specifically the SNP of RS11886868 in HbF levels and F cells. However, we did not see this significance in chromosome 6 and 11. And as I've explained in the previous slide, we're able to see the significance in males with high HbF as well as F cells with high, males with high F cells in these individuals. However, we're not able to see the same for the females. So to conclude our study, given the gender difference in distribution of F cells in sickle cell disease patients, we have studied the genetic association after gender sorting, and analyzing males and females separately may actually be key to finding associations for X-linked genes. This has yielded a positive result for fine mapping of long-sought X-linked determinant of F cell percentages, and these findings were actually validated. Our study has also highlighted the need for deep sequencing of targeted loci that might not have reached the GWAS significance in the previous studies. We can strongly now conclude by saying that apart from the 3 known loci that modify, apart from the 3 modifying genes that have been previously reported to play a role in modulating the levels of fetal hemoglobin and F cells, we have now added a 4th gene, a 4th candidate gene that may play a role in modulating the levels of fetal hemoglobin and F cells in sickle cell disease individuals. On the future follow-up studies, we hope to do replication studies in additional sickle cell disease population with larger sample size, and also we plan to make use of or benefit from the multi-country consortium, which is created by SICO in Africa and the H3 Africa, in order to design the GWAS array and bioinformatic analytic tools which will account for the African genetic diversity. We also plan to design a study to understand the mechanisms whereby a gene on the XP22.2 affect the gamma to beta switch, and therefore the number of F cells. By this we mean that we plan to do these functional studies using the erythroid cells in order to understand better the mechanisms of this particular gene. This work was published last year in October in the British Journal of Hematology. Lastly, I would like to acknowledge the study participants, the staff of MUHAS, specifically from the Department of Biochemistry and Department of Hematology and Blood Transfusion, my collaborators from King's College London, specifically the Red Cell Group, the funders of this project, the MUHAS CEDAR Small Grant, as well as the Commonwealth Scholarship Split Site, and my supervisors and mentors and all the co-authors of the paper for this project. Thank you.
Thank you. Thank you very much, Florence. That was fantastic. Every time I listen to your work, I really am inspired. Congratulations. And thank you to Professor Lucho for the supervision and mentorship. Thank you. So I think we will now, maybe Florence, yes, Florence and Heaven Light have turned their mics and Professor May is on. So we're happy to take any questions. So if you have any questions, please type them in the live Q&A. If you have any comments such as congratulations, could we collaborate, etc., you can type that also in the discussion forum. And so perhaps as a starting point, I'll ask Gaston to make any comments. But Aimé Lumaka, the work that you're doing in DRC is absolutely fantastic. And I think from the work that Florence has presented, as well as the work that we've had about pharmacogenomics in H3 Africa, I think there's a lot of potential collaboration. So I don't know whether you've had a chance to reach out to them and whether there are any opportunities that you may have learned during this this meeting. Gaston, do you have any comments or questions just to kick off whilst we're waiting for questions and answers?
Thank you, Prof. Julie. I would like first to thank all the presenters for great talk. And I think I have just one question for Prof. Aimé. So the title is so impressive. I saw pharmacogenomics, and I thought, oh, this is related to drug, and then you relate it to disease, pharmacogenomics of sickle cell disease. Why did you do that? And then the cutoff that you set for the cutoff of I think 15% for HbF. That you say, how did you set that cutoff? Because prior publication talked about 8.6% to improve SCD patient survival. Why did you set up a very higher cutoff of 15%? Why? And how did you get that 15%?
Yes, okay, thank you very much. So what we are, thank you very much, Julie, for the kind comment, and Gaston as well. For the comment and the question from Julie, actually, we are exploring ways to collaborate on the African level. We have not had great leads that we could follow and work on to join one of the existing network or the future networks, but we are open and willing to collaborate to the existing initiatives. For this particular study, this is a pharmacogenomic in a way that we are looking on the impact on genetic determinants on the response to a therapeutic agents. And the approach that we use, as I said, it's a candidate gene. We selected genes that had been associated to try see whether we can replicate this on Congolese patients. Previous studies, the one that we had before, which we had those 4 publications, we looked at patients and tried to classify them into clinical categories, whether those are clinically severe or clinically not severe, based on the number of crises they are having, vaso-occlusive crisis, based on the transfusions, and based on the presence or not of hepatosplenomegaly. We came out with a score, and when you look at those data, you would see that many of our patients, especially adult patients that were included in that previous study, they had a high level of fetal hemoglobin, which is sort of associated or explaining why they were surviving until the adult age. Some of the patients in that previous cohort, they were I would say reproductively active. They had 2 or 3 kids for women. So our cohort was in sort bias because these are the biggest chunk of the cohort we are working on. So for us, that came out as the threshold that would be interesting or that associated very well with either of the 2 categories, clinically severe and clinically less severe. For this study, we applied the same criteria that came from that particular study as patients from the previous cohort were included in this cohort, and we extended with children in the second setting. So it's a continuation, and then we continued with the same threshold of 15.
I think that's a great response to the answer, and your scoring system for disease severity is very, very helpful. Congratulations for that work.
Thank you.
There's a comment from Ambrose Owonkam. He says, great work. 2 of the significant SNPs are in known HbF promoting loci. I think that's the first bit is a comment and the second is a question for UMA. And perhaps you might want to link now with the discovery that Florence has made with the X-linked loci. you might want to include that in the 3 known reported loci that are associated with HBF production. So do you want to say anything about that? And then we have left, I think, 6 minutes. Then maybe we'll invite Florence and Heavenlight to say something, a couple of sentences before we close. Welcome, Eme, to answer the question from Ambrose or the comment.
Yes, actually the 4 of them, but 2 are associated with a strong significance.
Florence, do you want to make any comments, just general comments or something that you want to stress? And then after you, Heavenlight, welcome.
Julie, thank you very much. I would like to just thank. I would like to thank everyone for this opportunity to present some of my findings from my PhD work, and I'm ready to collaborate from any of the African countries that are present in this conference. So I'll share my email address, and we can talk more on the some of the work that I've done. Thank you
very much, Florence. Welcome, Heavenlight.
Thank you.
So Florence and Heavenlight are in the same office in Mumbidi, hence the echo. Welcome, Heaven.
I would like to thank everyone for their time.
Light.
Thank you everyone for your time to listen.
Thank you, Heavenlight. Sorry about the technical issues. I'll pass. Okay, unataka kumalizia. Do you want to finish saying what you wanted to say? Okay, Gaston, I'll pass the chair. Go on.
The internet problem. Just say a few words before my internet interrupts again. I would like to thank everyone for your time to listen to our presentation. We will hear more comments from you, and if any feedback or blessing regarding our site, please you are welcome to correct us. Thank you.
Very much, Heavenlight, and fantastic work. Back to you, Gaston, to finalize and close so that we can finish the session. Thank you, everyone, for your thoughts. Back to you, Gaston.
Thank you, Prof. Julie. Thank you to all the presenters for this session. It was a very great session, and we enjoyed it. And with that, we thank all the organizer committee and the African Society of Human Genetics for allowing us to follow all those great presentations for 3 days. That was very, very nice, and thank you for that. And I think with that we can just close and say bye. There is a session, the next session for closing the meeting, so please go to the next session so that we can hear last speeches from organizing committee and to close the meeting. Thank you everyone for attending this session. Bye.